and for coming and joining. So yeah, today I would like to talk about some of my latest works, which are on causality-inspired generative modeling and how we use them for studying diseases, specifically using single-cell genomics. So just to start by a brief introduction of this field. So our body has been generated from trillions of cells, and these cells are coming from different organs, and they have from different cell types. And so far, we have been annotating these cells into 400 different cell types, but this might even increase in the future. And these cells are different, and we want to know what are really the differences between these cells and how these cells makes different tissues and organs. But if we even broaden this perspective and look into different individuals, then cells are even more different. So because each of us have different genomes, and this makes even our similar cells different from each other. And some of us might have some disease or treatment background that this can also affect our cells. So the major question here is what are these cellular identity, diversity, and how can we uncovering them? So these in each, creature, then we have only one genome per cell, but this genome has generated so many different cell types. And we want to know what is actually the regulatory circuits that is underlying the cells and defines their identity. And then these cells together shape the tissue, and then these tissues, the organs. So all this process shapes a very fundamental question in biology to understand how different organs are shaped. And to answer these questions, single-cell technology has been quite revolutionary. So from a tissue, we select a sample, and then we can barcode each transcriptome in each of these cells. And then after sequencing, what we get, and is important for us as a computational person, is a matrix of cell by gene, where cells are the samples and genes are the measurements or the profiles. And in each of these, if we are sequencing transcriptomes, and then each of these items are actually the number of transcriptomes associated with that gene. And then we would like to find from this matrix the patterns between cells, the similar cells, and the different ones, using some downstream analysis, for example, dimensionality reduction, or some unsupervised methods for clustering them. So so this is what we are interested to do. But nowadays we have access to a large amount of single cells, and thanks to some large consortiums such as Human Cell Atlas that provides us with a lot of public data sets. And these statistics are from a year ago, so this might have been changed. But in the past, we had access to 50 million cells, that these cells were coming from 7.7 thousand donors, and they have been sequenced across different labs in the globe. And what makes these cells different from each other is sometimes the age and the sex of the patient. Sometimes it's the tissue or the organ that has been sequenced from them. And if they have been through some disease or treatment in the past, or if we have developmental samples, it can be the time or the space that can separate the cells or cause some sorts of variations in them. But these are some biological variations in the cells that we are very interested in understanding how they affect the response in the cells. But apart from this, there is a very important variation which is more associated with technical noise, and this comes from the platform or the laboratory that the data has been generated in. So this is something that we are not interested in, and often this is simply because there are two different people dissecting the tissue or doing the analysis, or maybe because of the different technologies that have been used for sequencing. So we are very much interested in being able to dissect the effect of these technical variations from the rest, which is more interesting for us. And then if we are able to do this, then our major objective is to learn from this heterogeneity in the data to go from heterogeneity in cells to heterogeneity across individuals. And also, when we can train such computational models with this data, we are more interested in being able to predict some of the effects of perturbations in the cells, which, for example, can be if a patient comes to a hospital, what would be the effect of this treatment in the patient can be predicted. And this is the basics of personalized medicine that we are looking into. So let's see how these learn and predict make sense and what they exactly mean here. So cells are very different from each other. Sometimes even morphologically, they look different and they have different profiles. And with single-cell technologies, we can profile these cells across donors, and then we can aggregate all these samples and build a so-called atlas of cells. And here I'm only showing you a two-dimensional representation of an atlas. And each point in this is a single cell, and it has been colored based on the similarities between cells. So one of the fundamental questions here is, what are these subtle differences between cells? So sometimes these differences can be the stochasticity in the biological process. So for example, the same neurons in my brain can just be different from each other because the microenvironment is different. Or sometimes it's because some part of the neurons might be affected by a disease or an injury, or it can just be a technical noise, as I said. So the first question, the first problem that we want to deal with is dissecting these sources of variations in the data. And then within each of these populations, we can then find the regulatory mechanism that is common across cells and differ them from the rest of the cells. This is another major problem here. But despite these tremendous efforts in this field for generating data, but still our data are limited by the number of donors and samples that we have. So, still considering all the human beings on the planet, then these atlases are just representing a very small, tiny part of the whole design space. So building, first of all, a representative atlas becomes very important, an atlas that can represent the whole population. But sometimes it might still be, with the current data, it might be impossible. So this is very important to be able to make the full picture or the full spectrum of cellular changes from only a snapshot of the data. So this is an artwork from my favorite artist, Gerhard Richter, that the artist has painted part of the picture and raises this question: what is hidden behind this? So this is the question that we would like to also answer with this type of technology and this available data. And when we can actually make these predictive models, then this can help us also to answer what experiment we should prioritize, because we are not able to do all the experiments, it's financially not possible. So we would like to know what experiment is more important. And this means that if I ask you, you're able to remove one piece of these colors to understand what is happening, which part would that be? So this is also the relevance to the biology for us. And to answer this question, I have been looking into generative causal models, and the generative part helps with predictability of the models. But with the causal part, I mean deriving some equations that explain the physics underlying the system, which can help us to predict the next state of the system. And this can, of course, improve the interpretability because we know how different variables in the system are working together, and also increases the identifiability because it derives the physical equations from the data. And finally, this is also some things that we want to be able to extend to biology by being able to predict the next stage of the cells. But to make this possible, we first need to learn a representative atlas. And then in the next slides, I'm going to show you how we have been addressing this and how causal models can help us to better learn these representative atlases. And then next, I will show you how this has improved predictability of the models as one example. So what we want to do is to learn a representation or a low-dimensional embedding of a multi-conditional data. And this means that the data is coming from different conditions or variant, for example, different ages, sex, or different diseases. And this is very challenging, and also it's called data integration. By integration, which just means that we are aggregating all these data. So this, I might use them interchangeably in the rest of the talk. And the challenge is that we have, let's say we have one sample which is coming, two samples that are coming from two different donors and two different batches, so two different experiments. And one of these, some cells have been affected by disease. And after the integration, what we would call an ideal representation of this data is a representation that, first of all, separates the major biological variations, which are the differences between cell types, but also within each cell type brings together similar cells. So here would be the blue cells from the both experiments. But this, because there is a minor variation also caused by the disease, we expect these to also be represented in the embedding. So we should be able to also observe the disease variations in the cells. So this model is called ideal because it has captured the biological signals, is invariant to noise because it has grouped together cells from different experiments, is interpretable biologically, and also is identifiable because if we run it with different initialization of the model, we always achieve this because this is the only way of capturing the biological signals from the data. But in reality, this is not what we often observe. So often we observe, for example, undercorrection, which means that the batch effect is so significant that even between the similar cells not affected by a disease, we observe a separation. Or sometimes we also observe overcorrection, which means similar cells are grouped together, but here this representation has masked the differences between diseases. So this is one challenge, but the other challenge is more about the application of these atlases. So let's say we build an atlas and we consider it as a reference atlas. The application then would be using this atlas for annotating a new data set. So in a new data set, we don't know the labels of cells. So we would like to be able to map these cells to the reference atlas and being able to annotate them. This is a very time-consuming process in single cell, so these atlases can be very helpful. But the challenge is that if this new data set has some new cell states, for example, here, these new blue cells, or they are slightly different, then many of these reference atlases might fail to properly map the cells to these reference atlases. And simply because these, for example, cells are unseen in the reference atlas, so it's probably going to map it on the most similar cells, independent of whether they are very similar or slightly similar. So this is another challenge that in our latest review paper, we have been discussing how causal models can help us to actually deal with these atlases. So the problem actually here is that many of these models that are used for representation learning are statistical methods, and these methods are actually they can also be based on spurious correlations. So here I'm showing you the interaction between variables in a statistical model, and many of these interactions might be just spurious and because of the correlation in the data. And the result is that if we use these statistical methods, they are very good in in training, in predictions in training environment, which means that if we train the model with different cells and different drugs, then if we test them on similar cells to the training data or similar drugs to the training data, then these models might be able to make predictions. But when we go to out of reference samples, for example, new cells or new drugs, then these models are very likely to fail in predicting the response of the cells to the drugs. But instead, if we have a causal model that the interaction between variables is very is based on the true physics underlying the system, then in this case, this model should be able to make the predictions also in this novel environment. But okay, this is a very ideal situation, ideal causal model. But to be able to derive these causal models, we also discussed in our paper that we need a diverse set of perturbations for training. And also we need enough replicates per perturbation to be able to train them. But apart from this, often in large problems, for example, in biology with thousands of variables, it's often not possible to have all the different diverse possible perturbations and have even replicates for them. So often what we can derive from a causal model for prediction is still limited to the data that we have been using for training. So here, if these points shows the data that have been used for training, our predictions, which in environment prediction is shown by green, and then our out of or the new environment prediction are the ones that have been shown in yellow. And there is still a large part of the designer space that these causal models might not be able to predict. But this generalization performance all depends on how much interventional data we are providing to these models. It can get improved, of course, with more data, but this is one of the limitations. Another challenge with this causal model is that often what we are interested in is finding the interaction between variables, the true interactions, and then to be able to infer this directionality between the interaction, the interventions can help. But then the problem is that these interventions are often assumed to be perfect. It means that we target one variable, we intervene them by setting them, for example, to 0, or in, for example, biology, it can be by knocking out the gene. Then in this case, this would help us to, for example, find some of the true interactions between genes. But in real cases, often the perturbations are not perfect. This means that we want to target, for example, G4 here in this graph, but indeed this perturbation is affecting other genes or other variables in the system as well. And this is what we call off-target effects. And in drug discovery, it has a lot. It's a big challenge as well. So this is also another things that we have to consider, that in real applications, sometimes having perfect intervention is impossible. So the question that now I would like to ask is, can we really then learn a true causal model of cells? And should we really spend time on learning it? Because it seems like we don't have enough data, and also we don't have perfect interventions. My perspective, which I would like to hear yours as well, is that currently with this amount of data and with the noise in the data, it's almost impossible to learn the true causal models. But still, we are able to learn models that are close enough to causal model, or at least inspired by a true causal model, by, for example, reducing the spurious correlation in the data. And this is what I call causality-inspired models. So they are not necessarily the true causal model, but they are inspired by this concept. And now I would like to go to one of my latest methods that I developed, which I think is trying to reduce the spurious correlations from the data quite significantly. So the idea is that we have control and a case cell, which has been, in the case, the cell has been affected by a biological covariance, for example, a virus. And this make a shift in the distribution of the response variable, for example, the gene expression. But what if I tell you that this cell has also been affected by, it has been sequenced in a different environment, so there is also a technical variations in the observation. So now the question is if this shift in the distribution is because of the virus or it's because of the technical noise or it's both of them, and often it's both of them. But how can we really understand which is which? The idea of invariant representation is that if we observe the cell in different environment under the same condition, then these environments are going to only affect a spurious correlation in the data. And the true relation between the disease and the cell should remain invariant. And therefore, using this assumption, we can derive the distribution of the cells, which can help us to dissect the invariant from the spurious correlations in the data. But how can we achieve this? This is the generative process that I propose, and I assume that my cells are generated from this process. And here X, variable X, represents my gene expressions, and I assume that my gene expressions in the cells are generated from two sets of latent variables. The first one is an invariant, and then the second one is spurious. The invariant factors are caused by are coming from a prior distribution, which is affected only by the biological covariance. For example, it can be disease. But on the other side, the spurious factors are coming from a different prior distributions, which is dependent only on the technical covariance, for example, the batch or the technology that we use. And we derive these prior distributions using the data itself. So these priors are inferred from the data. And then if we can learn these latent factors from the data, then we can use these invariant variables to derive the invariant representation, which should capture the biological variations across cell types and also across different diseases. And then we can also, more interestingly, understand what are the spurious correlation in the data. And this is very important because in most of the methods in the literature, there is no information about the spurious correlation, and they only provide us with the invariant. So we don't really know what we have removed from the data as a spurious. So with this, we can also visualize the spurious representation of the data, which should help us to find the technical variation between batches. And also it's another way of validating the results. And of course, there should be a partial independence between the invariant and spurious that we try to impose to the model by the right loss function. So in overall, the architecture looks like this. So it's a model based on variational autoencoder. It gets as input the gene expressions and the covariates, both biological and technical, and infers two sets of latent variables. But as I said, each of these sets have a different prior distribution that will be inferred and trained in the model. And then there is a decoder that learns the parameter of the distribution for the gene expression, which in our case is negative binomial distribution. And then our sample is reconstructed using these parameters of the distribution. And to see how this works in real example, here I brought you an example from the integration of or building a heart atlas of control and cardiomyopathy. And this is a very complicated data set because it has multiple diseases. We have control, we have dilated cardiomyopathy and arrhythmogenic cardiomyopathy. And also, apart from these, the samples are coming from different sex and age that in this particular data set, we are not interested in these variations, but we are interested in the variations caused by disease, and also we have different variants of these diseases. So PKP2 is the only one that is from ACM, but then the rest are associated with DCM. And also these cells are coming from different regions of the heart. We want to know also if these the anatomical regions of the heart impacts the cells and their functionalities. So to see the results, this is the first one, the first UMAP that I'm showing you is the representation of one of the related work that is widely used in the literature, and it's a very strong and nice method. It's called SCVI, and this method has been proposed mostly for homogeneous samples when the samples are coming from similar conditions. But here, I mean, I was challenging it by giving it different diseases. And what we observe is that, first of all, within each population, you can see that the cells from all these different conditions are merged together. This means that the model could find the major subpopulations in the data, but could not find the subtle differences across diseases. But indeed, we expect some differences between them. So this is the results of the INVAE, our method. And I'm highlighting here a population that is associated with fibroblasts in both of these representations. And what you see is that the control cells are nicely separated from the disease cells, and the disease cells are more similar to each other, which is also validated by the literature. And this is some benchmarking results that we use for evaluating the batch correction, which measures how well cells from similar conditions or from different batches, but similar conditions are merged together. And bioconservation, which measures how cells from different conditions, for example, control and disease, are separated. And overall, INVAE significantly outperform the related works. And this is because most of these methods that I'm showing you here, they have been proposed for homogeneous data sets. But this is just a two-dimensional representation of the data, and we want to know if they biologically make any sense. And for this, I dissected each of these cell types and zoom into them. And here I'm showing you the disease cell states only looking into fibroblasts. And the first UMAP is colored by the variants, and also the second one is colored by the disease and also my new annotations of the data. And the reason for this new annotation is that I noticed that there is part of the representation here in which some of the DCM cells are overlapped with the ACM cells. And these are two different diseases. So the first question was why we are observing these some DCM cells that are similar to ACM. I call them ACM-like. And then I wanted to know if this difference is also what we expect, what we observe in the expression of the marker genes, which have been introduced for these diseases. So in the first dot plot, you can see the expression of genes, some of the genes associated with these diseases for different variants. And as you can see, the 4 variants of the diseases are more or less very similar to each other compared to the control. And this is explaining why the control cells are very separated from the disease. But also looking into ACM and ACM-like cells, you can see that their expression of these markers are very similar between these two, but this is different than these DCM cells that are located here. This also, again, validate our results. And here in this bar plot, I'm showing you more or less the same thing, but here I just aggregated these genes and calculated an ACM score. And then you can see that also the ACM score is very similar between ACM and ACM-like cells. So with this method, we could find some new cell states in the DCM cells, which was not discovered in the past. So this is also even quite interesting for cardiologists. And the second example is about the prediction, so this is a generative model, but how does it work for prediction? And one of the ways to study this prediction performance is separating the data into training and testing data and make the predictions on that. So for this, here I'm showing you 3 data sets, 3 studies. So in this is also a cardiopathy or data sets, very similar to the one I show you. But the previous one was just only one study, but this is the aggregation of 2 more studies. And this is how I have also split the training and testing data. So in the training data, we have 2 studies, 68 donors, and this is how the cell types and the disease separation looks like as the outcome of the MVAE. And for the testing data, we had 2 scenarios. In one of the scenarios, the disease was very similar to the training data, but these are different donors and even different study. So there is batch effect, and also there is variations between donors. So it's a complicated, actually, prediction. But in the second scenario, which is even more complicated, we have introduced a new disease to the model. So this HCM1 has been even unseen during training. So let's see how the results look like. So this is because our atlas has both information about disease and cell type. Here we made a prediction also based on the disease and cell type. So we wanted to know if, for example, a cell is fibroblast, and if it's fibroblast DCM or is fibroblast healthy. So this is how we called each target, and on average, MVAE significantly outperforms some of the top methods in the literature for predicting the cell type as well as the disease condition from the cells. And I also told you about interpretability. Sorry. Interpretability and the fact that MVAE can improve that, because we are now also able to look into the spurious features or the features that we are removing after training. Because all the representations that I show you so far are only using the invariant features. So to test this, we had three classifiers, and for each of them we use a different prediction target. So in one case we predicted the batch ID using the latent representation, in the other one cell types, and in one other the disease. And we wanted to know what are the important features that the model might use for making these predictions. So so for predicting the batch ID, because this is an spurious correlation, we expect the model to only use the spurious features for making this prediction. And this is actually what we are observing, that the prediction of the batch ID significantly depends on the spurious features. And also for the cell type and the disease, we expect the invariant features to be the major drivers of the predictions. And this is quite nicely observed in the results. The last but not least results is how MVAE performs the second scenario, in which we want to predict the cell type, but for an unseen disease. So this is a very challenging task. But the thing is that here we can only predict the cell type, and we can't predict the disease, because the disease was unseen absolutely during the training. So this is why we are here we're only predicting the cell types, and predicting cell types is more easier than cell type dash disease compared to the previous one. And this is why even though still MVAE outperforms XGAMVI, which is one of the top methods in the field, but is it performing more similarly to SCP-poly and other methods for this purpose. But that's mostly because, I mean, this shift in the in the results compared to the previous one. As you see, the shift was more significant. This is because here we are not able to predict the disease, but if we could, probably in VAE would again achieves better performance. So in a nutshell, I mean, I show you some results of one of my latest methods, which is for integrating single cell data. And I told you that this model is inspired by causal models, and it tries to reduce the effect of spurious correlations from the invariant ones. And I show you with some examples that it's really learning the spurious correlations on some real data sets. And also it improves the cellular identification in multivariate data, which is a very complicated task in single-cell genomics. And we have even shown in our paper that we could identify new cell states, which is quite promising. And also with the last example, I show you how VAE performs for predicting the out of reference cell states and disease condition. So with this, I would like to acknowledge my colleagues from Fabian Theis' lab at Helmholtz Munich, and also my current group, Rosa Vento Tormo, as well as my collaborators, Joshua Bengio and Sarah Teichmans. Yeah, thank you very much for listening, and I'm looking forward to your questions.
Oh, thank you very much for a very interesting talk. Any questions? I'm sure you can, or either text or you can ask, and I'm gonna let you talk. I mean, this webinar allows me to have to accept talk, but I can ask. Yes, Chris.
Hi. Thanks for the talk. It's really interesting. So I had a question on the model, which you showed, I think, on page 16. You had the latent variables interacting. Could you say something a bit about that? And are they correlated or uncorrelated?
Yeah, thank you for raising this up. So this is some details that I just didn't get into it because of the time. But yes, the fact is that, well, in biology, we don't really know if the latent variables should be able to interact with each other or not. But we somehow guess that they should, because well, I mean, we don't know because we don't know what is exactly a latent variable. But if we assume that these latent variables are representing some sort of gene program activities or pathway activities, then we should expect them to also be correlated, because a lot of times pathways share genes and their activities can be shared. So in this case, we made in VAE flexible enough to also consider the dependencies between variables. But for this, we use general exponential family distributions for the prior on the invariant factors, so that we can make them also dependent. But in the examples that I show you, I'm often assuming that they are that there is no dependency, at least by I mean, in the loss function, this is what we assume, but I mean, in reality, I mean, if you calculate the dependency, there must be dependencies between them.
Okay,
just for the sake of simplicity, we did this.
Okay, thanks. That makes sense.
So we have a question in the Q&A by Olivia Gallup. She says, they say, thank you for the talk. Could you elaborate on how you treated the latent space for spurious and invariant variables differently? And also, why did you choose to use a binomial kernel for your VAA in compared to other kernels?
Well, yes, maybe I just very briefly share my slides. So for the first question, I mean, the major difference between these two is the prior distribution. So I mean, in variation on autoencoder, we assume that all the latent variables have normal distributions, mean 0, variance 1. But here we infer these prior distributions by, these prior distributions are different between the invariant and spurious. So for example, the prior distribution for the invariant is conditions on the biological covariates. So given that this is disease A, infer the parameter of the normal distributions for these variables. And knowing that this is coming from donor, for example, X, now also predict the prior distribution on the ZS. So that's how we elaborate between them. So why did we use binomial kernel? We used negative binomial distribution. We assume that the single cell data has negative binomial distributions. This is something that has been discussed in the field, and it has been shown that this improves the reconstruction and also the representation quite significantly. And that's why we infer the parameters of the negative binomial distributions and then predict the data from that. Reconstruct the data, sorry.
I hope that answered. Any other questions? I do have a small question about the methods, but Olivia Gallup says, thank you very much. I do have a question about the methods, just maybe a naive question, but so most of these treat when there's a difference in the gene expression for different biological conditions. So how would you try to study like cell apoptosis or when there's like cell death? I imagine because some treatments, kill cells. Or how would you enter that into the model? Like how would that be as an input?
Well, that's a very good question. I mean, here we do some pre-processing before giving the data to the model. So we hopefully remove all the cells that we assume that we think that they are dead cells. But still, maybe this is not properly, I don't know, filtered out. But something that's also relevant to your question maybe is that how can we add this type of information to the model in general? I mean, we can also have some other covariates, which I think can be only represented by some continuous covariates. So here we are only discussing categorical ones, but in some of these information, for example, if the cell is dead or not, maybe we can only explain it with some values that are not that are continuous rather than categorical. So in this case, yeah, we can actually change categorical to continuous, and this is something that the model can do, but we haven't tested it yet. And that's because going with continuous variables, it adds some concerns regarding identifiability. I mean, the search space increases quite a lot, and we are learning and embedding from each categorical variable. But in that case, we have to learn an embedding from the continuous variable. And then some things that we are very much interested in testing is if these make sense. For example, if sometimes we can't specify the disease by a name like ACM and DCM that I show you. Maybe there is a severity or there is a spectrum of a disease, and we want to assign actually a continuous variable to that. So these are all very, very interesting questions that we would like to look into in the future.
Beautiful. Thank you.
But that's interesting, yeah.
We have another question from another guest, Fabienne.
Hi, can you hear me?
Yeah.
Yeah, okay, great. Great talk. Thank you. I was wondering, kind of, and the last question kind of already hinted to that. I mean, in principle, what you could do with your model is also kind of style transfer, where you would say, oh yeah, what does this particular cell, what would it look like in this disease or in this specific context? Is that now technically should be possible, but does it actually work? Or have you tried doing that?
Well, this is what I'm trying now with some, because this requires experimental, I mean, the wet lab experiments. So this is something that we are very keen on testing. I mean, well, of course, you can, I don't know, in some cases with some synthetic data, you can show that I changed these cells and, oh, it's getting closer to the disease. But we want to be able to better verify these changes. So we are designing some experiments in the lab that that are based on the predictions of the MVAE. So if this answers your question, but I can't answer it now because we haven't tested it experimentally. But
Yeah.
I mean by just predictions, I mean, I don't know if I should trust just the predictions which are based on some synthetic, environment, synthetic settings.
Yeah.
So I don't feel comfortable answering that now, but in the future, I hope with some wet lab experiments.
Yeah, absolutely. But it just seems pretty straightforward. Wouldn't it be enough to just say, oh yeah, you remove a certain subpopulation from from your data set that you know exists, and that could somehow be represented by cell types or diseases or whatever, and then you see whether you find that again, or that you kind of can infer that from everything else?
Yes, yeah, we can do that. So yeah, at cell level, yes. And I think that the performance should be quite good because this is more or less similar to the predictions that we are doing. For example, when we are adding a new disease to the testing data, we are actually adding new cell states. So it's more or less, I think, similar to what you're proposing. What I was saying was perturbing genes, so at variable level. So that's more challenging.
Yeah, that makes sense. Yeah.
Yeah.
Thank you.
We have one more, another question by Chris Barnes. He asks, I have another question. Can you say something about what do you mean by the interpretability here? It seems you can see which latent variables contribute, but where do you go from there?
Well, yes, that's a good question. So interpretability, I mean, we can define it at different levels. So yes, you can say that what does each of these latent variables mean? This is a very open question in biology. I don't think anyone can answer this. We can always try to, I don't know, look for some enrichment scores, for example, associate these latent variables to some pathways. This is something that has been done in the past, but often still the results are not, because there is identifiability issue with many of these methods. These can change the results, interpretability of the results significantly. And this is why I'm avoiding it. And I don't think that anyone can really answer this question easily. But what I meant by interpretability is that we improve it by means of being able to remove the spurious from the invariant, and also showing that really the spurious is associated with what we call a spurious, with prior knowledge and some metadata. This is what I mean by interpretability. But yeah, that's a very good question, and it's very hard to answer at the moment.
Okay, I see. Thanks. Your response. Well, I think we are done with time. But thank you very much, everybody, for attending this webinar. And thank you especially, Dr. Hananeh, for your presentation and your talk. It was really interesting, and hope to see you all on our next seminar.
Yeah, sure. Thank you so much for the invite. It's my pleasure. Bye.